Today we're going to continue talking about probability and Punnett squares. And the goal of this video is to explain how to use a Punnett square in a dihybrid cross to predict the frequency of genotypes and phenotypes among offspring. In the previous video, everything that you saw was a monohybrid cross. And in this video, I want to focus on a dihybrid cross. Let me explain the difference between the two. A monohybrid cross is a genetic cross involving only one trait. Mono means one. So these are crosses that involve only one trait. These can be done using a 2x2 Punnett square, 2 columns, 2 rows. This type of Punnett square is what we used in the last video. All of the crosses in the previous video were monohybrid crosses. In this video, we're going to do dihybrid crosses. A dihybrid cross is a genetic cross involving 2 traits. Di means 2. And these can be done using a 4x4 Punnett square. There are 4 columns and 4 rows. I think it's helpful to use an example to see how dihybrid crosses work. So we can use fruit flies in this example because fruit flies have many traits. 2 traits in fruit flies are wing length and body color. Fruit flies can have long wings or short wings. Long wings are dominant. This is why the allele is represented with a capital letter, a capital W. And short wings are recessive. This is why the allele for short wings is represented with a lowercase letter, in this case a lowercase w. The other trait in fruit flies is body color. Fruit flies can be brown, which is dominant, represented with a capital B, or they can be black. Black body color is recessive, and this can be represented with a lowercase b. Okay, so that's just outlining some of the traits in fruit flies that we're going to use in our dihybrid cross. So here's the actual cross part. We're going to perform a dihybrid cross between a male fruit fly, this is the male, that is heterozygous for both traits. So to be heterozygous, you have to have one dominant, one recessive allele. Since there are 2 traits here, you're going to have one capital letter, one lowercase letter for the first trait, wing length, W's, and one capital and one lowercase letter for the other trait, which is body color in this case. So you're going to have, this fruit fly is going to have one big B and one lowercase b. This is a heterozygous genotype for both traits. We're going to cross this male with a female who is homozygous dominant for wing length and homozygous recessive for body color. So to be homozygous dominant for wing length, you have to have 2 big W's. To be homozygous recessive for body color, you have to have 2 little w's. So this is the genotype for the female. We're going to cross this male and this female and see what possible offspring they can have, what type of genotypes their offspring could have, what type of phenotypes their offspring could have. Now, to do this dihybrid cross, the very first step, when you're doing a dihybrid cross, the very first step you have to do is create the alleles in the gametes that are produced by each of these fruit flies during meiosis. So when this male fruit fly does meiosis, sperm get produced. And we want to know what types of alleles are going to be, or what combination of alleles are going to be in the fruit fly's sperm. The way that you do this is you use the FOIL method. That stands for first, outer, inner, last. Don't let that terminology throw you off. I'm going to show you how to do it here. So look at the male fruit fly's genotype. You are going to take the first letter for the first trait, which is a big W, and pair it with the first letter for the other trait, which is a big B. That big W and that big B are going to combine, and that will be found in one of this fruit fly's sperm. Now we're going to take that first letter, the big W, and put it with the other B. So now that big W and that little b, that's a combination of alleles. That goes in the second sperm. Notice we've put that big W with both of the B's, so we're finished using that big W for now. Now we look at the next one, the little w. we put the little w with the big B, and we put the little w with the other B. So these are the four types of combinations that this male fruit fly can produce, this combination of alleles in his sperm. All right, now we're going to look at the female fruit fly. Same thing, I'm going to go a little faster. So the big W goes with the little b. The big W goes with the little b. The other big W goes with a little b. Yeah, and that big W goes with a little b as well. So notice the female fruit fly, she really can only produce one combination of alleles. It's going to be a big W with a little b every time. In all of her eggs, it's going to be a big W with a little b. But this male produces sperm that have different combinations of alleles. This is just the first step in a dihybrid cross. You determine the alleles in the gametes produced by each fruit fly. Now you're going to take those gametes that were just produced and set them up in a 4x4 Punnet square. We have the female's eggs across the top and the male's sperm on the left side. And now we're going to combine those alleles inside each box. So the male gave a big W and a big B, and the female gave a big W and a little b. Those come together in this first box. Notice we're going to keep the W's together and the B's together, and we're going to keep those W's first. Now, this is the genotype this is a possible genotype for one offspring that this couple could produce. If this sperm and this egg came together, they could produce an individual with this genotype. What phenotype would this individual have? Since it has big W's, remember big W stands for long wings. That's the allele for long wings. So that's why we wrote long here. And since this individual has a big B is dominant, and the big B stands for the brown body color. So this possible offspring of this couple would have a phenotype with long wings and a brown body. I'm going to just fill in the rest of the box quickly, but if you'd like to pause the video and and check to see if you're understanding how to fill these in, do that now. So I've filled in the genotypes and phenotypes for all the possible offspring in this dihybrid cross. Now, usually you don't just stop there. We can begin, now that we've filled in this larger Punnet square for a dihybrid cross, we can start using that to predict the probability of a genotype or phenotype in the offspring. So take this question, for example. How many of the possible offspring are heterozygous for both traits? Well, to be heterozygous for both traits, you have to have a big letter and a little letter for both traits. So we're looking for boxes that have a big W and a little w and a big B and a little b. Those are the only boxes in this Punnett square that are heterozygous for both traits. That means that this couple has a 4 out of 16 chance. That ends up being 1/4 or 25%. 4/16 is equal to 1/4, and 1/4 is equal to 25%. So this couple has a 4/16 or 1/4 or 25% chance of having offspring that are heterozygous for both traits. Okay, here's another question that we could ask using this Punnett square. How many of the possible offspring have long wings and black bodies? I've highlighted those boxes that say long and black. There are 8 of these 16 boxes that represent offspring with long wings and a black body. Okay, 8 out of 16 is equal to 1/2, which is equal to 50%. So the probability of this couple producing offspring that have long wings and black bodies would be 8/16 or 1/2 or 50%. You could use any of those numbers. All right, that shows you how to perform a dihybrid cross. Now, I want to end the video by showing you a shortcut method. Rather than making that large Punnett square, the 4x4 Punnett square, in this shortcut method, you can actually just use 2 Punnett squares that are the 2x2, the smaller ones. And each of those Punnett squares represents the traits. So, and to finish this off, you will then multiply the likelihoods. So let me show you an example to make this clear. We have the same couple of fruit flies right here. This is the male, this is the female. They have the same genotypes that we looked at a moment ago. Here's our question. What's the probability that this couple will have an offspring that is heterozygous for both traits? If you recall from a minute ago, we said the probability was 1/4. Or we said the probability was 4/16, which is equal to 1/4, which is equal to 25%. Okay, I want to show you how to get that answer using the shortcut method. Here's how you do it. You take the first trait, wing length, and you're going to make a Punnett square for just it. So this male has a big W and a little w for wing length. So we're going to put those letters in the sperm. And this female has 2 big W's. Those are the alleles for wing length in the female. So we're going to put the 2 big W's in the eggs at the top. Now, when that sperm and that egg come together, they make big W, big W. I'm just going to fill in the rest of this Punnett square. Okay, now we're going to look at the other trait. So we have another Punnett square for the other trait, which was body color. This male has a big B and a little b. So those go in the sperm for the male, big B, little b. This female has little b. So those go in the eggs for that female. And then we fill in the Punnett square just like normal. Okay, now look back at our problem. What's the probability that this couple will have an offspring that is heterozygous for both traits? So look at the first Punnett square. How many of the possible offspring are heterozygous for wing length? These 2 individuals, 2 out of the 4. 4 possibilities here. 2 of those 4 possibilities are heterozygous. Okay, what about the other Punnett square? There are 2 out of the 4 possible offspring that are heterozygous for that trait as well. This is where, now that we know in our first Punnett square, 2 out of 4 are heterozygous, and in the second square, 2 out of 4 are heterozygous. This is when we're just going to multiply those likelihoods. So 2 out of 4 times 2 out of 4 equals 4 out of 16. 2 times 2 is 4, and 4 times 4 is 16. So the likelihood is 4 out of 16, the same answer that we got a moment ago when we set up the larger Punnett square, the 4x4 Punnett square. And remember, 4/16 reduces to 1/4, which is equal to 25%. So there's a 25% chance that this couple will have an offspring that is heterozygous for both traits. This is a shortcut method. You can use whichever method you like, but I showed you how to do a dihybrid cross using that larger Punnett square and how to do a dihybrid cross using this shortcut method.